welcome everybody. Today, my student Stefan Popp is defending his PhD thesis. This is a slightly old picture of him. I want to say that Stefan, I think, distinguished himself in my lab not only for extraordinarily complex analyses and huge data sets, but also what I thought was really interesting and fun is with Stefan in the lab, we had very rigorous discussions about everything else in life, from the Pomodoro method to how you balance frustration and progress, and many other things. We started a book club. We read about grit and time management for mortals and sort of how to optimize every aspect of your life. And Stefan's always taken that very seriously, I think, more than any other student I've had in the past. So it's obviously bittersweet to see him go, but I'm also very proud of what he's achieved. And I want to show you just a couple other pictures. So I don't know if you can recognize, this is at an alumni dinner, which was incredibly fun and made me look very old. These are all people who were alumni from my lab at a conference, and Stefan is there. So that is early in his career. And then, I put this in. This is from the Insect Festival, but it's going to show that Stefan also always participated in a lot of the outreach we've been doing and took that seriously. And then this is the most recent one where we taste tested some ants in an ant lab in Israel, a conference we were both invited to. And if you compare the first picture to the last picture, you can see that the hair migrated around, but the shirt is still the same. So with that, I'm looking forward to Stefan's presentation.
All right, thank you, Anna, for that lovely introduction. Fun throwback. So I will, jump right in to my thesis about the ant search strategies for unknown targets. I will be going over the 4 chapters that are part of my thesis, and I will go over the meandering pattern, familiarity, coordination, and finally a chapter on resampling. Okay, so I will jump into the first chapter on meandering. So first of all, to set you into the right scenario, imagine that you are on a wide meadow like this one, and this is about as big as the campus of the U of A, and you're tasked with foraging or finding those lovely little mushrooms there. And every time you find one, you have to come back to your home base to attack. What is your strategy? Maybe you're thinking, well, I should search in a systematic pattern like in this spiral here, or in a pattern like this, like a meander or a zigzag. Now, I want to go over these 2 different ways of covering an area in a search. So one is the systematic that we just heard about, or the other one is a random walk. So the question is, how systematic should we actually search in a real world scenario? That is because in the mathematically ideal world, it's clear that we should in a systematic pattern, because in a random walk, you're always covering the same areas twice where you have already searched and you know that there is nothing. But in the real world, where you also have to contend with navigational noise or noise in your locomotion, or also obstacles in the way, that scenario becomes very different. And here, maybe the systematic way of covering an area is a lot worse than the random walk, because that kind of noise that you encounter is already basically baked in to the random walk. There is a study that actually shows this: with high enough noise, systematic patterns become worse. So what do animals do? Well, we have assumed in the literature that animals walk or search in a random walk pattern, and this is also how they have been analyzed all the time. But I want to know whether that is actually the case in the ants that I studied. When I say that the ants are searching, then I mean they search for something like water, or new nest sites, or competitors, or maybe potential invaders into their nests, or of course for food. First, I will go over the general methods of the first 3 chapters that are my empirical methods, so they are all the same. I studied the rock ants, Stemnothorax rugatulus, which, as the name suggests, nest in rock crevices. They are relatively submissive and slow ants. They show no trail recruitment. So unlike the other ants that you might be more familiar with, they have not these strong recruitment trails. So that means that the foraging success of any colony is defined more or less by the search success of every individual, and that goes out and searches for her own. They also deposit footprint pheromones, and we assume that they are passively deposited by just the ant walking over a surface. And there are also some pheromone glands in the feet that facilitate that. So we get and colonies up from the Catalinas in a habitat like this. We suck whole colonies in, and then we put them into artificial nets like this one here, 50 to 300 animals per colony. And then I put the whole colony inside this arena, and I track the ants with 4 cameras and automatic tracking software for 5 hours each. So I have 5 colonies, 5 hours. This is a huge arena here, undergrads for scale, 2 by 3 meters, which is about 1,000 times the length or the size of an ant. And in the end of one trial, this is the kind of data we get, a lot of tracks. You can see in the right where the tracks are coming out of, that is the location of the nest. And there are a lot of data there in just one trial. All right, so coming back to the question, what do ants do? Do they search systematically or randomly? Well, I show that ants do search systematically in a meandering fashion. So on the orange trail here, that is a random walk, and the black one is the real ant track. And so how I figured out that they are actually doing these meanders, and this is not just the product of some random characteristic, is that I first created these random walk null models, of which I showed you one, and then I measured the turn autocorrelation. So I created the random walk null models, about 2,000 of those, by just shuffling around the individual tiny steps that I get from the tracking software, such that the general characteristics of how squiggly the ant is moving around should be the same, but any long range turn autocorrelations should not be in those null model random walks. So with that, I can then compare it, the autocorrelation that should only be there in the real tracks and not in the null model tracks with one another. And so if the turn autocorrelation is 1, then the ant is always going into or turning into the same direction. If it's -1, then the ant is doing a perfect zigzag. And if it's 0, then there is no correlation between the turn directions of any successive steps. And here what I find is on the x-axis are the 1,200 ants or something. And on the y-axis, the lower, the more meandering the ants are. In green, all the significantly meandering ants. So 80% of the ants do meander significantly more than we would expect if they were just doing a random walk. And they are doing this on multiple scales. So if you consider here one meander, then there is layered over on another scale, another meander, basically. And then we can go one more step down. And this is now finally the actual part of the track of one ant that was just walking around. What I find remarkable here with the specimen is that it does never cross its path in this maybe 2 meters that the ant is walking around in real terms. And this is also the case for a lot of the ants. In fact, 83% of ants cross their own paths, significantly fewer than we would expect under a random walk. And then here, the higher up on the y-axis you go, the less the ant is crossing itself compared to the random walk. So that means that this meandering is probably a very efficient strategy in order to avoid crossing the areas where you have already searched. This is maybe also the reason why you can find it across the tree of life between bark beetles, moth larvae, Dictyostelium, which doesn't even have neurons or a brain, and to the bottom left next to the meme, there is Paleomeandron, that is more than 500 million years old, a trace fossil of just a worm that was gliding around on the bottom of the ocean. So that means that this pattern or this mechanism is probably a very ancient way of moving around and covering an area very efficiently. The thing is that not many people have investigated how animals are actually doing this and in which ecological scenarios they are doing this. This is something I would like to further investigate, maybe in the coming winter. So in conclusion of the first chapter of the meandering, the standard assumption of animal search is that they are doing a random walk, but this is not the case in my ants, which are doing a meandering pattern, most of them. And this meandering and self-avoidance is done on multiple scales. And that is probably also a lot more efficient than regular meanders or regular patterns only, or only randomness alone, because it combines the efficiency and also the robustness to external noise by also adding some randomness. And this has been published, yay. Okay, so now we'll go over to the second chapter. So how does your strategy change when you have already learned where you are at this meadow? After maybe the second or third day, you know already where the best mushroom patches are. And if you're far away, you know a lot better how to get back to your home base, to your tent. This is a very ecologically relevant scenario for these Temnothorax that I study because they move their nest a lot. And that is because they don't build a lot of their own nest, so they don't like build into the grounds. But here you can see in red outline, this is the only thing that they're building. And when that nest is open or disturbed, then they will just move to another nest site. They also move to improve. So if there's another nest site nearby that is better, somehow bigger or darker, then they will also readily emigrate to that one. And when they're doing so, they also move closer to our resource-rich areas. They also split colonies seasonally. So in the spring, they split their colonies into multiple nests. And then in the fall, they consolidate back again into the same nest site. So that means that they move multiple times per year. And so whenever they are moving, it is important for them to familiarize themselves with the new environment around the new nest site so that they know where the potential risks are or the resources, and that they can find back home to the nest. And so I studied how they change their search or their exploration behaviors over consecutive days. So I filmed them not only over one day, but same colony over three consecutive days, 5 hours each, and no resources in this arena and nothing else. And this is visually the results on the first day. Again, the nest is to the right, to the bottom. You see that they're clumped around the nest, their exploration. On the second day, it looks a little bit different in that they are a lot more spread out. And on the third day, you can see they're still spread out and there's a lot more white in between. So there are fewer ants, fewer ant tracks out there. And I can also show you that in figures here on the x-axis, the days, and on the y-axis, the distance walked in kilometers for each colony. So there's less exploration activity with increasing familiarity. There's more even exploration with increasing familiarity. So here the colored lines are the medians of each colony of how far away they on average move from the nest. And they are significantly different. So they are moving further away with each day. Ants are also walking straighter with increasing days. The numbers are relatively the same, but we have a lot of data. So these are all highly significant. What I mean by straightness, or how I calculated this, is a local straightness. So I take between 2 points on the track, I slide those 2 points across every track, and I take the Euclidean distance between those 2 points, and I divide that by the actual walked distance by the ant. And so this gives us a value between 0 and 1, with 1 being almost a straight line, and 0 means that the ant has been doing a loop between those 2 points that I took on the track. Ants also walk straighter within days. You can't really see that well, but again, they are significantly becoming straighter and also faster. I don't show you the data for the speed because it's in the same direction, but they move faster and also straighter, which are correlated with one another. I also show that this is not driven by any footprints that I introduced you to, because what we did was between the day 2 and 3, we exchanged the paper overlay. And with that, we removed any chemical footprints that the ants could have been depositing. So if they were only reacting to the footprints, then this is what we would see, that the trend reverses between the first and second day to the second and third day. But as I showed you, we don't see the trend reversal. So they are still increasing between the second and the third day. So they're not reacting to the removed footprints. In conclusion of that part, with increasing familiarity over days and also within days, there is less activity and the ants are moving farther away from the nest, exploring wider. They're also moving faster and straighter. And this is not only driven by footprints. There are at least three non-mutually exclusive hypotheses. One is that in the beginning, the ants are patrolling for risks, maybe for other intruders that might want to steal their brood closely around the nest, or they are memorizing their surroundings in order to know where to go and how to come back home. Or maybe they are reacting to resource scarcity, so that over the 3 days, they know that there is no food to be found, so they just spread out and fewer even try to get food. All right, so this brings me to the third chapter. Now, imagine that you are searching a search team with maybe 10 or 30 other individuals that are on your same team, and you somehow have to coordinate, but you can't really talk directly to them for whatever reason. What would you do? What would your strategy be? Maybe you pay close attention to the footprints that your search mates are leaving in the grass so that you avoid those areas because they have already picked all the mushrooms there. And this is obviously very relevant to the ants. As I have shown you already, they deposit footprints passively. And I investigate how or whether they are changing their behavior, their walking behavior compared to the footprint concentration by calculating this proxy of a footprint concentration. So it's not the actual chemicals concentration there, but it's just an adding up of, imagine an ant is at some point at some place, then I count up how many times another ant has already passed this area where the ant can basically taste with her antennae where other ants have been before. And so previous work has actually shown that ants cover an area around their nest more evenly when they are walking on footprints, so when other ants have been walking on there before. So that means that they do have to coordinate somehow. And I investigate two hypotheses. One is that the ants are turning away from the side with a higher footprint concentration. And the other one is that the ants are moving straighter when they are on the footprints. So the first one is moving away from where others have already searched to find new places. And the second one is when you are on a patch of where others have been searching already a lot, then you want to go straight out of that area. And then you can walk more tortuously outside of that area. We do not find that ants turn away from the footprints. And you can see that in the plot here on the x-axis is the difference of concentration between what they can sense in the left and right antenna. And on the y-axis is the actual turn angle that is correlated with that. And because you can see that there is a completely flat line, there's no slope, that the ants are not turning either towards or away from the side of the higher footprints. Okay, but what about the second hypothesis, that ants move straighter when they are on footprints? This is also not the case. Ants, in fact, move less straight on footprints, as you can see by the negative slope here on that plot. So the more footprints, the less straight they become, which is kind of counterintuitive. That got us thinking, why is that the case? Why do we see this opposite correlation of that second hypothesis? Now, the first obvious, the reason for the less straight movements on footprints is twofold, but it's not a biologically interesting correlation, but more springs out of the math, that there are fewer footprints farther away from the nest, which is certainly the case. As you can see here, the farther away from the nest, the bluer, so the fewer footprints there are in the arena, which is just logical from geometry. And at the same time, there are some ants that are moving straighter, and those straighter moving ants, they move farther away from the nest. This is also expected if you look at mathematical random walks. So this leaves us then with this scenario where the less straight moving ants are staying close to the nest and just happen to stay also on higher footprint concentrations. So if we plotted this with, on the x-axis, ants starting out straighter, going more to the right, and on the y-axis, the higher it goes, the farther the ant actually goes in the arena. So we would see a positive slope there, and that's what we do see. So yes, this hypothesis is true that straighter ants move farther away where there are fewer footprints. So that means that there is this causation leading from the straightness to the number of footprints instead of the other way around, which we initially wanted to look at. So that means that if we are controlling for this effect of the distance to the nest, then this correlation should either vanish or it should lessen or it should even flip. And so to test for that, just to be sure, we binned the data by the distance to the nest, and then we ran the same analysis of the correlation between what is seen on the y-axis here, between the straightness and the number of footprints. So if the values are 0, which is the medium line there, then there is no correlation. So the ants do not move any differently on higher versus lower footprints. If it is negative, so on the bottom one where you see the plot as well, then you see a negative slope. and on top is the positive slope that would more be in line with the initial hypothesis. What we see is all negative slopes. So again, this cannot explain how ants are covering the area more evenly. And it might still be that footprints actually make ants walk less straight, opposite to our expectation. But then this got me thinking, maybe this is not the whole story. And again, we have some correlation that we didn't think about. Maybe it's that some areas in the arena, regardless of the distance, make ants walk less straight and thus deposit more pheromones there. So what I mean by that is if you look at that horizontal line there that has relatively low straightness values, that is where there was this adhesive tape, Scotch tape, just 2 or 3 layers of paper beneath the surface layer where the ants are actually walking on. So that means that they can smell through these multiple layers of paper where there is something that they don't like. And so they bump into it, they go back. And so that makes them walk relatively non-straight. And that if you squint your eyes a little bit, then you can see that there are some a bit more footprint pheromones on the strip. And so to test for that, I binned the whole arena into 2 millimeter by 2 millimeter pixels. And I tried to disentangle the effects of the characteristics of the pixel from how the pheromones might influence how the ants are walking. So on the x-axis, it's basically the characteristic of how straight or non-straight this pixel makes the ant walk. The measurement is that the straightness of the first ant that crosses this pixel. I just take that to be this characteristic, and on the y-axis is the final number of footprints per pixel. And this negative slope tells you that this is indeed the case, that some areas, so those where the ants are walking straighter to begin with, they will accumulate less footprints, which does make sense. So that is another explanation. Again, moving into the direction from the straightness of the ants, causing a different number of footprints and not the other way around. Again, so that means if we are controlling for this effect of the pixels and the heterogeneity making the ants walk differently, then we should also see that this correlation, again, of this negative slope should vanish or reverse. So the y-axis is, again, as I showed you before, and again, we see negative values. All the asterisks are significant ones. And so that again means that even if we are controlling for this, that negative correlation still persists. That means that finally we can be relatively sure, also because we repeated the analysis on another data set of this original paper up there, that the footprints actually do make the ants walk less straight. We didn't really know yet why this would be the case. But in conclusion, there's more even area coverage, and this is probably done more through inter-ant variation, that some are straighter to begin with, and not direct communication through the footprints. There's more even area coverage also through large avoidance of footprints or large scale. So this is the hypothesis of how they can still react to the footprints, but maybe it's more, 20 minutes ago, I've been in an area of really high footprint concentration, and so this is why I want to stay away from it. And importantly, there is an extreme sensitivity to environmental heterogeneity, especially in terms of the smells of what they're walking on. And this is much more important in the natural habitat than the chemical footprints that we assume that they are leaving behind, which are just minuscule in comparison. Okay, last chapter. This last chapter is a little bit different, because I only did this on the computer. It's nothing empirical, and it is a methods paper. And I was glad that I planned on this not being empirical, because that's what my arena looked like last year at the end of summer, where a bunch of sewage just spilled out and I could not take any more data. I was okay, because I was more concerned with the fact that when I was analyzing the data, then I was either using the turn autocorrelation or the local straightness, which just considers the whole tracks or all of the points on the track. But in studies of animal movement in the ecology, we're oftentimes more interested in discretizing the track so that we can have an easy analysis of that. So we need to discretize the smooth or noisy tracks into steps and turns. And where one true turn is a biologically significant one, so that means where the animal has intended to move or to turn into some direction. And this is not noise where the animal just maybe due to wind or locomotory inaccuracy, the animal was just veering away from the course. And you can see a very clear example of that with this bacteria in the top right that have relatively straight steps and then turns in between. But it's, of course, not always that easy. Because if we take a look at this example here of an ant track, how would you identify significant turns here? Maybe like this, so that you get straight turns, or maybe like that. Those are different types of tracks, and we don't really know what is the better one, and maybe they also have different characteristics. So how do we make this discretization objective? The solution to that are resampling algorithms. So those are also called rediscretization or turn identification methods. How they work in general is that for all the points in the track, you do this algorithm until a certain threshold is met. And the threshold is something that you define as the user. I have two examples. One is the non-local one that deals with the raw angles. So you start with a point there, for example, in this case, to the left, and then you add up all the turn angles, and you add them up until you reach the threshold angle that you have set before. So the green angle is still okay, and then the red angle is already beyond the threshold. This means that you only keep the point before the summation of the angle was beyond the threshold. And then you continue with that throughout the rest of the track until you have discretized the whole track in this fashion. So you will end up with only relatively large angled turns. The other one that I want to go over is the Douglas-Peucker method or algorithm that deals with distances. So here you create straight lines between 2 points, and you only keep that point which has the largest distance to that straight line. So in this case, it would be the second circle from the left. And then you iterate over the rest. So you repeat that until you're left with only distances that are below the threshold that you have set in the beginning. And so in this case, you end up with the same track in the end, which is kind of lucky. But there are a lot of algorithms that have been proposed, more than 10 in the ecology literature. And I call it a bit of a siloing going on there, because how you select which one you are using is probably what I have the intuition is mostly guided by who you cite or who you collaborate with, who does the thing in your field. So one example is with primates here and the change point test. There are more than 15 or 20 papers that actually use this change point test, and only 2 of them do not use primates. And as far as I know, all the people that are working on primate movement, they also use this change point test. I have never seen anyone actually say that this is objectively the best test for this scenario. And so others don't even know about this test, as far as I know. An analogy in evolution that I've conjured up is that when you're building phylogenetic trees, you're also having to choose which method you're using, and then which program you're using, which parameters, which genes, and so on. So that will also change your phylogenetic tree probably, and which one you use is also not very objectively determinable. So the problem is then which resampling algorithm is the best. And there are 2 aspects of this. When they are proposed in the literature for the first time, there's oftentimes not a really comprehensive validation. So, and this is still a good example where they have 9 simulated tracks that they test this against, but oftentimes it is much less. And there are also very few comparisons to other methods. So in this example here, they compare their new method with one previous method. So this is 1 out of maybe 8 or 9 that have come before this method was published. And so maybe others are still better than the method that they have published in this paper. And when they're being used, then the thresholds and the methods that are being used in the empirical papers, they are oftentimes not well justified or compared even to other methods or thresholds. For example, here they write that this method is similar to another one and has been used by another paper, which is not really a good justification that is actually the best method that they could be using in this example. The solution to that is my 4th chapter, where I compare these resampling methods. So how I do this is I can't just take a real animal track, because we don't know where the intended turns are that the animal was making. So we have to make ground truth tracks. And the way I did this is by lining up a bunch of points on a straight line, then connecting a bunch of straight lines with these points by turn angles. And then I added noise to every one of these individual points so that there are some large turn angles that are true turns and a bunch of noise that are now not that straight lines anymore between those turns. So those are the ground truth tracks, and we know the characteristics of these. Then I resample a bunch of those tracks with the resampling method that I included, and then I compare them with the metrics, with lots of metrics, but I will only show two of them, the number of turns and the mean turn angle. And those are metrics that are also oftentimes used in the field to characterize or compare tracks. So now I'll go into just a little bit of detail. Here is a sample of the ground truth tracks that I created with different parameters and different noises. And I have 100 replications for each parameter set. Yep. Then on these tracks, on each of these, I run the resampling method. So here in black is the ground truth with a bunch of noise. And I will just add in a bunch of these other resample tracks on the net ground truth track. And you can see that they're all somewhat different. And the question is now, does this difference matter, or do they have the same characteristics pretty much? Therefore, I compare them on metrics, on the number of turns, and also two other metrics that I will show. That is on the y-axis, relative metric. So 1, which is right there, would be the perfect value, because this is matching the ground truth track perfectly. If it's above 1, then the resample track is overshooting in that, for example, here it would be number of turns and vice versa. On the x-axis, there is going to the right, the ground truth track is becoming more squiggly, more tortuous. And what you see in the results is that there are a bunch of colored lines, which are the different resampling methods. Some of them overshoot by maybe a factor of 2, and others undershoot in this number of turns by maybe a half. And also within some of these resampling methods, there's a large variation within the method. So depending on which track maybe of an animal you have, you're going to get a different result as if you were using a different track of the same animal that has the same underlying characteristics, which is, of course, or can be a problem. And so also between different metrics that you're comparing, there's also variation. And what I want to point out here is the one in red, that is the non-local that I introduced you to in the beginning. So it's pretty good in estimating the mean turn angle, but it's not so good in estimating the turn angle standard deviation. So how much the ground truth turn angle deviates from the mean. And so this could also be potentially important and difficult to tease apart which one is now the best method for your specific application and what you want to find out. Then I was also interested in the susceptibility to noise. So here in the different panels, there are different ground truth tracks of more squiggly to the right. And on the x-axis, the more to the right you go, the noisier the ground truth track becomes. And what you can see here is that these lines are all more or less flat, which is surprising because that indicates that the more noise there is, it doesn't change the accuracy of your resampling methods. It kind of all stays the same. And also, if you go to the right in the panels, the noise to signal ratio becomes better, but these resampled tracks also don't become better in their estimation of the mean turn angle in this case. One reason for this might be that I cherry-picked the results from the best thresholds. For each parameter set, I only saw, okay, maybe 0.6 is the best for the local method. And then I only presented these in the results, which is very generous to the resampling methods. But this is, of course, a problem in the real world, where what people are actually doing is either having an educated guess about which threshold they use for the method, biological knowledge, so saying, maybe, we expect the animal to only be turning in 60 degrees, even though we don't know at all. Or they're doing it in a post hoc way and saying, okay, with a 60-degree or 90-degree angle, we see the best signal in our data, or it fits our distribution the most, which is obviously also problematic, possibly. And these thresholds is also what I see. They matter, and sometimes they matter a lot. Again, if we just take as an example a non-local method, if you have the threshold at 20 degrees, then you're overshooting by almost a factor of 2. And if you set the threshold to 30 degrees angle, then you're undershooting by less than half. And so this is potentially also a problem. But wait, there's more. there's a lot more than 100 methods of resampling that have been proposed in the human mobility research. This is an excellent review. And in the human mobility, they're called curve simplification, and they're used on commuters or traffic or freight shipping. And they have much more influence from computer science, this is what CS stands for, and math. So they are basically in different bodies of literature. This is maybe also why we in the ecology literature don't really know about any of these, as far as I know. And so this means that we should probably consider those, because we don't know whether any of these are possibly a better fit to our animals, to our scenarios and problems. So in conclusion, this is probably an issue for comparability between movement animal papers, and it's also a problem for reproducibility. A different method can lead to different conclusions of a paper. So you always have to justify your methods and thresholds, and that also goes for, of course, the methods that you're using in other fields of biology. Once this paper is all fleshed out, I will hopefully give recommendations because some methods are better for on certain scenarios or metrics that you're interested in. And new methods that are being proposed should always be comprehensively evaluated with the ground truth data, as I'm doing here, and compared against many already existing methods and not just one or two that seem convenient to you. And of course, we need a lot more interdisciplinary and inter-system communications also between different study systems, for example, as this opinion paper in the bottom right suggests. On that note, this is all that I had to say about my research today. I would like to thank everyone who made it possible. First and foremost, my advisor, Anna Dornhaus. She's the reason that I came to the University of Arizona, and it's been a good decision. Of course, I have learned a lot from her in terms of how to do science and also thinking about things deeply. It has been always a lot of fun, but she also made sure that we're all well taken care of, that we're all okay. And beyond that, I felt really like I have gained a second mom in her in my time here. So it's kind of painful to leave the lab, but I'm also proud that I will join the illustrious web of alumni of this lab. And I am looking forward to still being in contact with her. To come down for that a little, I would like to thank the funding as well that allowed me to live this lavish lifestyle of a PhD student. Also thanks to my committee, who have always given me great insights and comments and asked the hard questions. Tom Kennedy on the bottom left, I unfortunately had to replace because he's currently in hospital in France. But thank you to Camille, who just jumped in. And it's nice to meet you. I had a lot of fun collaborating outside of my PhD thesis with, for example, people in math and computer science, trying to actually figure out what the ants are doing when they are meandering left and right. Like, what is the algorithm that they're going through? I also had the opportunity to go to Western Africa to do some field work on the super cool ants, the metabelly ants. You can totally not see them, but this is a raid of maybe 1,000 or 2,000 ants that are currently going out to break open some termite nests. And I was interested in what the scouts are doing and how they are finding the termite nests. Then also, I think a 4-year collaboration on and off with these people from a workshop on the mathematical biology that resulted in a paper. Thanks to a long list of undergrads who painstakingly cleaned the data and worked through plots like these in order to connect all the tracks that are poorly connected from the tracker. I'm also grateful to people in the lab that have been great mentors and friends along the way. And my cohort, first and foremost, Noah and Adam, who are some of my closest people. I love you all. Other EB grads, when I came in to the 6 years ago to the department, I just realized that the most salient memories I have are from the senior grad students who became kind of role models to me. So I hope that I had a good influence on the more junior members of this EB department as well. The staff has always been really cool and helpful. One of the things that were most salient when I came here to the United States, that the people in the office are usually really like happy and they seem like they enjoy doing their jobs. And so it was always great seeing happy faces in the offices, and also my friends in Germany who I could keep in contact with more or less. And of course, my family in Germany. Danke an die Familie. die mich immer unterstützt hat in meinen sonderlichen Ausgängen und die es letztendlich auch akzeptiert hat, dass ich einen anderen Lebensweg eingeschlagen habe, weit weg. Die wöchentlichen Englischklassen über Skype mit meinen Eltern und die monatlichen E-Mails mit meiner Schwester waren eine konstante Quelle von Freude in meinem Leben. I have to translate from English to German. And with that, I go back to the science, and I like to thank you for coming out in the middle of summer, and I hope that you could take away something from it, and I'm open to any questions. Yeah, so we do know something, not in this species, but in others. The composition is partially from the glands, but also a lot of the cuticular hydrocarbons. So they are just basically shedding them. The problem is that those amounts are just really tiny, and we could possibly find out what they are in the species and then recreate them, or maybe just strip them off maybe dead ends. The problem is always with those pheromone studies that maybe you have too much or too little of a certain compound, and then even if you don't know, but it's because the ants are so sensitive, as you know, you might not be able to get like a good surface maybe of the footprint. So in theory, it is possible, yes. I did not mark the ants, so I don't know between individual tracks of them leaving the nest and then entering, which one is the same ant of another track. I can't say whether there are any differences, but I can say that there are, within each track, there are some differences. Maybe some ants are straighter near the nest or less straight near the nest. Yeah. And then between the tracks, there are these differences that are, at least within one foraging or searching bout, that they are relatively consistent. I also see that. Yeah, it all depends on what you're actually interested in how you characterize the tracks. But for this example, the standard, the variance of the turn angle, if you only assume, coming from your methods, that the animal is always turning at 60 degrees, then you're going to miss maybe the influence that some signal or some cue from the environment might have that still lets the animal turn, but only for 10 degrees or 20 degrees. So you're missing a lot of that, those correlations, the influences that might be biologically significant. I just started with setting up another arena that I wanted to use for another experiment, maybe someone else will take it over. And I started 3D printing a surface that has like bumps in it. And I'm really excited about this because they have to change their behavior somehow. Either they are trying to cover the area that is like on a three-dimensional surface as if you were spreading it out, or they care more about how far away they get from the nest. So then they would have to go straighter, but in the third dimension, they would still be relatively tortuous. What I have found in very preliminary experiments is that the ants are moving more tortuous. So they are actually not moving as far away from the nest because they're just searching basically the area that they can perceive more tightly. What it did not present is that maybe the animals are not actually walking in steps and turns. So for example, when an ant is turning, she's actually doing an arc. So it might be that there are no steps and turns, but the ants are continuously integrating with continuous time, continuous space. I'm not sure how much we know about how it works on the neuronal level, but there could be a good connection in that if you resample the track in the right way, you might be able to get the best fitting path integration. Yes, but we don't know how the ants are actually, or how animals are deciding how much to turn and in which way. So these ants, the study species varies by a little bit, maybe 10% of the size, but also in other ants vary a lot more. Between species, at least, larger ants move, they have different stride frequencies than smaller ants, and it might change the pattern a little bit. I'm not sure. It could be that the smaller ants are doing, for example, tighter meanders. It's a good question. I have not thought about this.
All right. Well, thank you, Stefan.